Hi everybody, it's Tim Shelberg coming to you from New York City with my co-host Vanessa Lynch for the 9th annual DNA Hit of the Year.
Thanks, Tim. I'm excited to be here and proud to be part of GTH DNA's Hit of the Year. This program is all about real-world impact, solving crimes, bringing justice, and reuniting families through forensic DNA.
The cases we're sharing today highlight just how crucial DNA databases are, not just for identifying suspects, but also clearing the innocent and finding the missing. This year, we received incredible submissions from around the world, showcasing the latest advancements in forensic DNA. After careful review, we've selected 5 finalists, one of which will take the title of DNA Hit of the Year.
Our 5th place case takes us to our Tukey foothills just outside Phoenix, Arizona, where rapid DNA played a critical role in solving a brutal triple homicide. At the crime scene, investigators found a blood sample near the victims and initially suspected it belonged to one of them. They sent the sample to the Phoenix Police Department's Rapid DNA unit. Within hours, the unit generated profiles for all 3 victims and the unknown blood sample. The results were clear. The blood didn't belong to any of the victims. It likely belonged to the killer. Investigators quickly entered the profile into Arizona's DNA database, and almost instantly they had a match, a known offender. Arizona is one of 7 states in the United States that currently allows crime scene evidence tested on rapid DNA to be searched against the state's database of criminal offenders. With this lead, police swiftly arrested the suspect, who then led them to an accomplice. This case is a perfect example of how rapid DNA helps law enforcement take dangerous criminals off the streets in record time.
Incredible work by the Phoenix Police Department under pressure, Vanessa. Cases like this show how rapid DNA is transforming investigations, and it's only going to grow. Starting July 1st, new FBI standards will allow crime scene DNA tested with rapid technology to be searched against the National CODIS database, which holds over 22 million criminal profiles. This is a game changer. Law enforcement agencies and crime labs across the United States are already preparing for the shift, and soon, rapid DNA will be a standard tool in solving crimes nationwide. Our 4th place case highlights the growing use of YSTR databases to solve crimes when standard STR searches don't get a match. In 2022, a 21-year-old hiker in Malaga, Spain, was shot and killed during a robbery. Investigators found DNA on his backpack, but when they searched Spain's National CODIS database, there was no match. Fortunately, Spain, like a growing number of countries, allow investigators to compare crime scene YSDRs against a YSTR database of known offenders. When they searched the YSTR database, they got a hit. Not to the killer, but to a male relative. That lead helped police quickly track down and arrest the murderer. It's a great outcome, but it raises a big question. Why wasn't the killer already in the Spanish STR database? It turns out he was a convicted killer who had been released from prison in 2013. Even though Spain had DNA database legislation in place back then, the legislation didn't allow retroactive DNA collection from prisoners. Spain is known for having one of the best managed DNA databases in Europe, and the National Police did an outstanding job using every available tool to solve this case. But it also shows the limits of DNA database laws. If the killer had been in the system from the start, the case could have been solved even faster, and there wouldn't have been the need to move to the YSTR strategy. Later in the program, we'll take a closer look at how lawmakers can ensure DNA databases are as effective as possible in delivering justice and keeping communities safe.
That's right, Tim. The National Police of Spain did an incredible job solving the case, but their work was made harder by outdated legislation. Our 3rd place case also required thinking outside the STR database, this time using forensic genetic genealogy, or FGG. In 1957, the body of a 4-year-old boy was found in a cardboard box in Philadelphia. He had been severely beaten and malnourished. The case known as the Boy in the Box became one of the most infamous unidentified human remains homicide in US history, puzzling investigators for over 65 years. Finally, in 2022, the Philadelphia police office of forensic science identified the boy as Joseph Augustus Zarelli using FGG. With its name restored, the homicide investigation has new momentum, and there's hope that his killer will finally be identified. Since the Golden State Killer's arrest in 2018, the world has seen how FGG can crack some of the oldest and most tragic cold cases. Initially used to identify suspects, it's now becoming a powerful tool for identifying human remains. With so many unidentified victims around the world, FGG is critical when government DNA databases don't provide answers. Every victim deserves to be identified. Mike Garvey, Director of Forensic Science at the Philadelphia Police Department, submitted this case for Hit of the Year, summing it up perfectly, and I quote, "Advances in forensic science give us capabilities that were once impossible. These breakthroughs, along with the dedication of our detectives and scientists, will ensure no future victim remains nameless or forgotten."
Thanks, Vanessa. There's still so much untapped potential in using DNA databases and forensic genetic genealogy to identify human remains. We're only beginning to see what's possible. Now, let's move to our second place case, a disturbing serial rape investigation from South Africa. The offender was linked to at least 49 attacks, making it one of the most significant cases of its kind. Being from South Africa, Vanessa had the chance to speak directly with the crime lab experts and investigators who worked this case. She's got valuable insight into how they finally tracked down the suspect.
Tim, this case is a perfect example of how DNA isn't just a tool for solving crimes, it's a game changer, especially for connecting serial offenders when there's no immediate suspect. It also highlights why strong DNA policies and a well-maintained database are essential. Between 2012 and 2021, Nkosinathi Phakathi terrorized the Ekurhuleni Metro in South Africa's Gauteng province, attacking women and girls as young as 9 years old. He struck early in the morning or late at night, targeting victims on their way to school or work. Sometimes he even posed as an electrician to gain entry into their homes. For years, Phakathi remained undetected. His attacks seemed random, with no clear pattern linking them. That is, until DNA changed everything. In 2015, South Africa passed a landmark DNA law, creating a national DNA database and requiring that every arrested or convicted individual provide a DNA sample. Just as important, it mandated the testing of thousands of untested sexual assault kits, ensuring crime scene DNA was uploaded into the system, even without a known suspect. Once police could compare profiles across cases, they linked 11 rapes from 2012 to 2014, and later another 37 from 2015 to 2021. That's 48 cases tied to the same unknown suspect. Before DNA, these crimes were not even connected. Even with those links, they still did not have a name. So in 2020, a special task force was formed, comprising of detectives, forensic experts, and investigative psychologists working together to reanalyze the case. They re-interviewed victims and built a psychological profile, uncovering chilling details. Phakathi stalked the victims, studying them before attacking. He was highly methodical, carefully planning his crimes. When victims resisted, he sometimes pretended to feel guilty, even asking for forgiveness. With these new insights, investigators identified 49 additional cases with the same pattern, bringing the total to 97 known rapes. Now they just needed to find him. At one point, another man was wrongly arrested after being misidentified in a police lineup. DNA proved his innocence, a critical reminder that forensic science isn't just about catching criminals, it also prevents wrongful convictions. Knowing Phakathi was still out there, police urged victims to report any sightings. In March 2021, they got a tip. A schoolgirl had been approached by a man near a small local convenience store. She recognized him instantly. He was the same man who had attacked her before, but he didn't recognize her. She reported it immediately. The next day, police set up surveillance in the area. When Picati returned as expected, officers spotted him and matched him to the profile that they had built. As they tried to arrest him, he attempted to escape but was shot in the leg. At the hospital, a buccal swab was taken and DNA confirmed what investigators already knew. They had their serial rapist. With overwhelming evidence, Picati had little choice but to plead guilty. If South Africa hadn't prioritized testing old sexual assault kits and expanding its DNA database, Picati's crimes may never have been connected. Worse still, he could still be free, hurting more victims. This case proves that it's not just about having the right technology. It's about laws that support DNA databasing, skilled forensic experts, and strong collaboration. When all of those come together, DNA becomes an unstoppable force for justice.
Thanks, Vanessa. Incredible case. It's clear that prioritizing DNA databases and casework is the key to solving crimes like this. Okay, it's time to discuss our top case. The 2025 DNA Hit of the Year goes to a case that remained unsolved for nearly 5 decades, a brutal crime that left investigators searching for answers across generations. A breakthrough finally came in 2023 thanks to advances in forensic DNA and a key change to state law. What followed was an extraordinary investigation that connected 3 police departments and revealed a truth hidden for nearly 50 years. To walk us through this incredible case, we're honored to welcome Rock Harmon, co-chair of the DNA Hit of the Year. A former Alameda County prosecutor, Rock was among one of the first in the United States to use DNA evidence in criminal trials, shaping the future of forensic science.
In 1975, Hallie Seaman, a promising graduate student, was murdered in Seattle. Her case quickly went cold, joining countless others in the archives of unsolved crimes. For nearly 50 years, investigators searched for answers. Leads dried up, DNA evidence profiles sat in CODIS unmatched, and time marched on. Until 2023, when everything changed. Then a long overlooked DNA offender profile was collected, one that had never been entered into any database. The reason? The person it matched had been executed long before DNA collection from deceased offenders was required. After decades of uncertainty, police finally had their suspect and the evidence to prove it. Thanks to Jennifer and Michelle's Law, a recent legal reform in Washington state, a long-standing gap in the DNA database was finally closed. Named in memory of Jennifer Bastian and Michelle Welch, 2 young girls brutally murdered in Tacoma in 1986. This law was enacted to ensure that DNA is collected from deceased violent offenders who had never been entered into the system. Before its passage, deceased criminals who had never been swabbed for DNA while alive remained outside forensic databases, leaving crucial connections undiscovered. With this law in place, investigators were able to upload a long overlooked DNA offender sample into the system. And the result? An immediate match. The Hallie Seaman case is a story of determined investigation, significant legal reforms, and advanced forensic science that finally brought long-awaited answers. At the heart of it all is Charles Campbell, the ruthless predator who committed the crimes and who was executed in 1994. Today, 3 key experts who played a vital role in solving this case are here to share their insights. First, we have retired Snohomish County Sheriff Rick Bart, who was the first officer on the scene of Campbell's brutal 1982 triple homicide. Next, we have Lindsay Wade, a former Tacoma PD detective, author, and criminal justice advocate. While investigating the 1986 murders of Jennifer Bastian and Michelle Welch, Lindsay uncovered a major flaw in the DNA system. Dangerous criminals like Campbell weren't even in CODIS because they had died before the effective date of their law. She led the fight to the legislature that brought about the changes that solved the case. And finally, 30-year veteran detective Ralph Norton of the Seattle PD, whose passion for solving cold cases played a crucial role in reopening the Hallie Seaman case, using forensic advancements to solve it.
Hi, I'm Rick Bard. I'm really glad to be here. I'm the retired sheriff from Snohomish County, Washington. It's a county that's just north of Seattle. And for 12 years, I was one of the two Snohomish County Sheriff's Office homicide detectives. We were busy, and I pulled the Charles Rodman case.
Thanks for having me. My name is Lindsay Wade, and I am a retired detective with the Tacoma, Washington Police Department. I spent 21 years with the Tacoma PD and spent the bulk of my time in investigations, primarily homicide and missing persons and sexual assault cases. And working on cases involving DNA really has been my passion throughout my career. And so I'm just glad to be here.
I appreciate being here. Thank you for having us. My name's Rolf Norton. I'm a Seattle Police homicide detective. I've been with the Seattle Police Department for 30 years, and the last 24 years, I've worked homicide. When I first began in law enforcement, my ambition was to investigate murders. There's no higher calling, there's no greater purpose. We chase down what society deems to be the ultimate evil. In 2017, I reopened the Hallie Seaman homicide case, and the journey that occurred for the next 5 years was incredible.
Rick, you were first at the scene for that 1982 triple homicide. Can you tell us what your first impressions were when you got there and explain how the investigation unfolded?
Sure. I was the first detective there. I had a partner who lived a little farther away, who was on his way. I didn't go in the house yet. I needed a search warrant to do it. And so I started writing that. And finally, when my partner got there, I had already learned that one of the victims lived across the street from a patrol officer. And so he went over to talk to the family while I finished the search warrant. And once I got it, I went down to the house, around to the back, because we decided that's where we'd go in because the sliding door was already open. And that's where the patrol people had gone in. So that's where I went. I went in quickly to see what I had. I went down the hall and stopped, did not go in the back bedroom, and then came back out and waited for my partner. And once he got there, we came up with a game plan, what we were going to do, and we started our crime search. We've been to a lot of homicides together, bad ones, and this is the worst one we ever saw.
So you mentioned that one of the victims lived across the street. While you were there, did you learn about a connection between that victim, the two victims in the house, and his prior crime?
Yes, the patrol sergeant that was there he used to be a homicide detective for Snohomish County, and he talked to me about investigating a rape a few years before, and that one of my victims in my house was the victim of that rape, and another victim in my house was the neighbor lady. And he mentioned Charles Campbell's name and said he's the individual who was convicted. He was supposed to be in prison. And so I asked him to go find him, see if he's in prison so we can get rid of him and go on our business, eliminate him. And so he did. He took off to try to find Mr. Campbell.
So 2 of your 3 homicide victims were involved in his prior conviction for rape that he had been sentenced to prison for.
Yes. Not only that, when he was convicted for the rape, he stood up in open court and told the judge he was going to kill 2 of the victims when he got out of jail.
Given his prior history, how long a period did he serve time for that earlier rape?
Not long enough.
Do you remember how many years?
I think 7, 9, something like that, 7 years.
Okay.
That's about the time that the sergeant came back to the house that I told you about earlier to tell us that he'd found Mr. Campbell in a work release in Everett, which is the county seat in my county. And what did we want to do? And I told him to go back up there, call his parole officer if you can, and see if there's any way you can find a way to keep him detained because we need to talk to him. It was difficult for us to control our emotions. When I finally got to talk to him, he was taken over to the Monroe prison and put in a cell there. I went up there, I think the next day or day after that, with a search warrant for his body and brought him out to talk to him. And very hostile, very angry guy. The head of a guard in the room with us and wouldn't talk to me, of course. Everything just fell into place. It just was boom, 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 boom. And when we found out that he had been let out of jail, he came through in his promise when he was in court. He told the judge, I'm going to kill him.
Lindsay, your work on the murders of Jennifer and Michelle opened your eyes to some very big problems in Washington State about that hampered your investigative abilities, and they concerned DNA and CODIS. Could you tell us what you learned that caused that concern?
Sure. It's actually kind of a long, convoluted story, and it started well before I was investigating Jennifer and Michelle's cases. So I have to go back to 2010. And that's when I first discovered that there are convicted rapists and killers out there who are not in our DNA databases. And at that time, I was working on a cold case, and I was on the phone with someone from the Department of Corrections. And just sort of off the cuff, I asked a question about the Special Commitment Center in Washington State, which is held on an island. And it basically, the only thing that takes place out there is it's a detention center for sexually violent predators. And these are individuals that have been convicted of sex crimes, and instead of being released after they serve their prison sentence, they can be civilly committed indefinitely. And so during this conversation on a totally unrelated matter, I sort of accidentally discovered that there were about more than 40 sexually violent predators on that island that had never had their DNA collected. And when I started making inquiries and asking why, and asking for the samples to be collected, I really got nowhere. And actually, I got a lot of pushback from the state.
What kind of answers did you get at first?
Initially, I was told that the policy was to collect the DNA upon release. And so, don't worry, detective, we'll get that DNA, before we release them. Which is completely ridiculous because, number one, a lot of these people will never be released. They'll die out there. And many had died out there, 9 to be exact, that never had their DNA collected. And also, what about the unsolved cases? Do we want to sit around and hope and that maybe detectives could solve these cold cases another way? So it was just a completely misguided way of thinking. I eventually got the Attorney General's office on board, and they called out to the Special Commitment Center and apparently lit a fire under somebody. And they finally decided to start collecting the samples. It took almost 2 years, which is completely insane that it took that long, but 2 years to swab 41 people. And we did have a couple people refuse to give DNA, and so we had to get court orders to get them to be compelled to give DNA samples. But ultimately, those samples were collected, they were sent to the CODIS lab, and they were eventually uploaded. And as a result, the 1980 murder of a 19-year-old woman in Bellevue, Washington was solved because the offender who murdered her back in 1980 had been sitting out at the Special Commitment Center since 2001, and he was completely forgotten about. That case from 1980 was sort of my first peek into that world, and as time went on, I started finding more and more examples of these really horrific offenders who were not in CODIS. And so the offender from that 1980 case was one. Ted Bundy was the second person that I stumbled across that wasn't in CODIS. My partner and I were working on a cold case from 1961, which was our oldest cold case at that time in Tacoma. And it was thought by many that she was potentially Ted Bundy's first victim. So she was an 8-year-old girl who was abducted right out of her home in the middle of the night. And so going down the investigative road on that case led me to ask the question about Ted Bundy's DNA, and sort of like the people at the Special Commitment Center, the answer was no. His DNA is not in any database. And so that was another long project, working with the Florida Department of Law Enforcement to finally track down a sample of his DNA, which had been collected in 1978. and it was sitting in a safe in a clerk's office in Florida, a blood vial that had never been refrigerated or frozen. So you can imagine the state of the DNA sample. But miraculously, the lab was able to develop a complete profile for Ted Bundy, and it was uploaded into CODIS finally in 2011. So those were like my two initial cases that I learned about. And then from there, I sort of started taking a different approach when I was investigating cold cases that had DNA. Cases where we had a profile, but there was no match in CODIS. And the two cases that you mentioned earlier, Jennifer Bastian and Michelle Welch, were exactly those kinds of cases. These were two little girls that were murdered in 1986 in Tacoma. They were 12 years old and 13 years old, respectively. Both were blonde-haired, blue-eyed, out riding their bicycles in Tacoma city parks. Both were abducted and sexually assaulted and murdered within two different parks about 1.5 miles from each other. The two crimes occurred within about 4.5 months of each other. And because of all the similarities, this is well before DNA testing was the norm, everyone thought it was the same killer.
Mm-hmm.
And so early on, once we had DNA in the first case, we thought, okay, DNA is definitely going to be what solves these cases. A few years later, we got DNA on Jennifer Bastian's case, and we learned we had two separate offenders in these two cases. But nevertheless, they were still unidentified. And so those cases, they were voluminous, thousands of tips and leads and named suspects and potential perpetrators to go through. But it made me think that, one of the investigative strategies that we need to pursue here is looking at who's in prison already. Like, who has history for this kind of behavior in whether it's Pierce County or some other area in Washington State, and who's not in CODIS. Let's start there.
Right. How easy was that to determine?
It wasn't easy at all. Trying to identify who's in CODIS and who is not is extremely difficult. And so what I started with was reaching out to the Department of Corrections and asking for a list of convicted offenders who had convictions for rapes or murders, kidnappings that were not in CODIS. And that is where I first heard the name Charles Campbell.
Lindsay, you've described the troubling things you learned about who is not in the DNA database. And you also described your reality that Charles Campbell was not one of them. How did that motivate you to fix things?
I had this massive list of convicted rapists and murderers on my desk that were not in CODIS, and Charles Campbell was on that list. And so that's how I first even heard his name. And on a list with several thousand people, the reason his name jumped to the top was because I could see that he was convicted of aggravated first-degree murder ×3, and in the status column of the spreadsheet, it said executed. So that was very unusual. And given the fact that I'd never heard of this guy and I'm from Washington State, I took it upon myself to do some research and start looking into him. And what I learned was horrifying.
So these discoveries must have been a pivotal moment for you in trying to change things. So what steps did you take to initiate the reforms that we know you achieved?
Yeah, basically I would just sort of keep track of example after example of what I called buckets of criminals who somehow slipped through the cracks and were not in CODIS for a variety of reasons, either because they were convicted before the law went into effect, or because the law just simply doesn't address them. There were lots of reasons why I was uncovering these issues. And so at some point, I had the opportunity to advocate for legislation. And I had a state representative named Tina Orwell who came to my agency and wanted a tour. And she had a lot of questions for me about working cold cases. And I developed somewhat of a relationship with her. And when it came time for the the legislative session, she was very instrumental in not only sponsoring the first bill that we tried to push forward, but advocating down in Olympia for this DNA law. And it wasn't a quick process. It took 4 years before this law was finally passed. I would put together a PowerPoint presentation, and I'd have some clear examples of individuals who really needed to be in CODIS but were not because of the way the law was written. And so Charles Campbell was one of my examples. So I literally had a booking photo of Charles Campbell and a description of his crimes, going back to the basically home invasion sexual assault of his original victim, who was home alone with her infant daughter, going to prison, getting out after a very short period of time with no notification to the victim. Victim had no idea that he was even out of prison. And then him coming back and committing this horrific revenge murder on her and her 9-year-old daughter and her neighbor. When the law was finally passed, it did several things. It added the crime of indecent exposure to the list of gross misdemeanors that requires a DNA sample to be collected, which I think is very critical, because a lot of times people, think that indecent exposure is not that serious of a crime. But if you look back on most rapists and oftentimes sexually motivated killers, they will tell you that they partook in many, many episodes of indecent exposure. So that was one addition. We also were able to broaden the crime of refusal to provide a DNA sample to anyone who is required to provide DNA, not just registered sex offenders, as it had been written previously. And then the third and most important addition to our DNA law was the fact that law enforcement could now get DNA from deceased convicted offenders and get that DNA into CODIS. And prior to that law change, if the person was convicted, even if they were convicted of a violent crime, a rape or a murder or kidnapping, if they were convicted before the law went into effect, they did not qualify for CODIS. And so Charles Campbell was, a perfect example of that. He was a rapist. He was a convicted killer ×3. He was executed, but he didn't qualify to be in CODIS because he was convicted before the DNA law went into effect in 1990.
Sure. And those are all three pretty important components, because we don't know who we're going to catch with DNA,
right,
unless everybody that should be in there is in there. So Lindsay, to tie things together as we've been discussing them, the legislation finally passed in what year?
2019.
But the two cases, Jennifer's and Michelle's, had been solved before that, and you stayed on to make sure the legislation passed.
Yes.
Now, there's another step there, too, because somehow Charles Campbell ended up in a database.
Yes.
And I'm sure you're going to tell us the role you played in making sure that happened.
Yes. So, kind of going back to discovering Charles Campbell's name in the first place and seeing that he wasn't in CODIS, years earlier, I had started actually doing some research to try to see if I could locate a sample of his DNA. And I knew that he had an autopsy, and I reached out to the medical examiner's office to see if there was any evidence, and there was. So I got really excited. And then I called the crime lab, and they told me, that's great, Lindsay, but you can't put Charles Campbell into CODIS. He doesn't qualify. So that's how we ended up down this road of trying to advocate for Jennifer and Michelle's law and using him as an example. Now, once the law passed in 2019, I went back to the medical examiner's office and got a sample of tissue that had been maintained by the medical examiner from the autopsy, and hand-delivered that to the State Patrol CODIS lab in Seattle. And that was September of 2020. And that was the last I heard of it, and didn't hear Charles Campbell's name again for 3 years, and honestly forgot all about him. I just assumed that there were no hits.
So Lindsay, I hope you realize at this point, you did a lot more than change the law. You paved the way for future law enforcement to solve their cases by including more people in CODIS. And for that, I'm sure you're appreciated and remembered. So thank you very much.
Thank you.
Rolf, you worked on the Holly Seaman murder case for years, and you experienced one of those magical breakthrough moments when at least the DNA had been identified. Can you take us back to that moment when you learned that information?
Well, let me go back a little bit further, because the story comes to a conclusion in August of 2023, but the story begins in April of 1975. Hallie Seaman was a 25-year-old graduate student at the University of Washington. She was getting her master's in architecture. One Tuesday night, she went to work in her architecture loft on campus. The student that she worked with saw her there at 9:40 p.m., and she left shortly thereafter, never to be seen alive again. Her body was found the next afternoon by an individual taking a break at work. He went outside to exercise. He saw something in the bushes. This was underneath I-5 off of East Lake. And he investigated further. He walked towards what he was looking at, and he saw it was the body of a young female. Her dress was in disarray. She was partially closed, laying in a very unnatural way. Became a full homicide contact, and they soon learned that she was stabbed 9+ times. She had been sexually assaulted and discarded at that location. Her vehicle, a white Ford Fairlane, was found a couple miles south in the Soto district of Seattle. It had been torched and left on the train tracks. It took a couple days to identify the victim. So the body was found, but the initial investigators didn't know who she was. Her sister had reported her missing the day before, but the link or the nexus had not been made yet. It actually was her professor called the homicide unit and said, "Hey, I have a student who shows up to class every day, and she hasn't shown up for 2 days, and she kind of matches the description of the body you found." And boom, they found out it was Hallie Seaman.
Wow.
So during the initial autopsy, forensic evidence was recovered from a person that ended up being the tool that got the case solved. Years went by, the case got put aside. Until 2002, we had the opportunity to go further with forensic analysis involving DNA. And our autopsy evidence was submitted to the Washington State Patrol Crime Laboratory, and a suspect DNA profile was identified. Now, we don't know who this person is, but we have this person's genetic code. And if anyone at that point says this case was cold, it wasn't cold. It was old, it was unsolved, but there's our suspect. The genes of our suspect. We just need to match a name to that person. And that's where things got challenging. There was 120 names in the murder book, all vetted, many cleared with direct comparisons. Case didn't get solved. I picked it up in 2017, did a full review, and started from scratch. The next year, the potential of genetic genealogy broke, and we put together a list of the Seattle PD of cases that potentially could be addressed with using this strategy. And Hallie Seaman's case was at the top of the list. So we requested a reformatting of the DNA recovered from the victim. However, it was mostly consumed, meaning we didn't have enough product available to reformat it to an SMP file. So the case goes on. And at that point, I was just looking at every name and retracing and retracking and going and talking to people and just hoping for a stroke of luck, hoping to hit a lottery ticket. And then in August of 2023, I got an email from the Washington State Patrol Crime Lab scientist, William Stubbs, who did the initial analysis in 2002. He put the case number on the subject line. He said, "Rolf, I'm going to be sending you a report on this case. We should talk about it. I think you're going to be surprised." I'm looking at the case number and I recognize it to be Hallie's case. And I called William Stubbs and he says, "You holding on?" And I said, "Surprise me." He says, "DNA matched Charles Campbell." And he let that sit there. And I remembered Charles Campbell because he was executed right around the time I was hired by the Seattle Police Department. And I remembered that he had horrific sexual assaults in 1974, that he served 7 years in prison and got out, and he went back and killed everyone involved in that case, including the victim's 9-year-old daughter.
At that point, did you realize that a law change had facilitated or authorized the state to collect his DNA?
I did not. So one of my first questions to Scientist Stubbs was, why are we talking about this name in 2023? Because his conviction for sexual assault was 74, his murders were 82. Why is the name coming up now? He referred me to the director of HIDS, as he said, it's kind of a longer story. And we then started talking about the SAKI initiative in 2019 that Lindsay was working on, giving law enforcement the latitude to take offenders that offended prior to 1990 and enter them, or at least search them in HIDS. After I spoke with Scientist Stubbs in August of 2023, and he shared with me the name of the offender, one of my next calls was to the King County Medical Examiner's office, because my understanding was that his DNA was collected from their autopsy, ironically, after he was executed. And talking with the pathologist there, he said, yeah, talk to Lindsay Wade. She was involved in this. And I said, Lindsay Wade? I know Lindsay. And shortly thereafter, I reached out to her and said, hey, I got some news for you. And I told her I had a 1975 case of 25-year-old UW student who was killed, and we matched a profile. And his name's Charles Campbell. And Lindsay's reaction was, you gotta be blanking kidding me. And I said, no, I'm not blanking kidding you. And we went back and forth on that for like 5 minutes, because she was all on this name, because it had been on her mind. I also called the Snohomish County Major Crimes Unit after we received notification. I was talking with now retired Detective Jim Scharf, and he said, Rolf, you need to call Rick Bart. He was ultimately our sheriff, but he was the original investigator in the 1982 triple murder. So tracked down a phone number and called him up at home. And it's probably a long silence when I say, hey, this is Detective Rolf Norton from Seattle PD Homicide. There was a long pause. Like,
What the hell is Seattle PD calling me for?
And then explain the scenario. And Rick had a long pause hearing that name.
It was an old crap moment. And I thought about it for a few seconds, and I still couldn't believe it. But I knew DNA was a great tool and was really changing the way we investigate homicides. But when he told me that, and I thought about it, it made sense, really. But then I thought, darn, if we had this stuff back in 82, my victims would be alive, I would think, because he'd have caught them. And he'd have taken care of the problem. So that kind of angered me. But I think this whole thing gives, most importantly to me anyway, is families that have to go through this and lose a loved one. It gives them hope. And I've seen it in their faces. And we've solved some cases that we couldn't before because of DNA. I've seen them lost hope. And then you call them and tell them, and it's back.
Yeah.
That's wonderful.
It gives me great pleasure to give each of you the Hit of the Year Award for 2025 for your contributions, all of you, in the Hallie Seaman case. Forensic DNA isn't just a technological advancement, it's a lifeline for justice. By closing outdated legal gaps, we ensure that no case is too old to solve and that serial offenders can't be safe in prison. The Hallie Seaman case is a reminder of what's at stake and why we must keep pushing for change. While states may have variations in the DNA laws, the single most significant improvement would be to collect DNA from all eligible offenders retroactively. I'm Rock Harmon. This is how changing the law can finally bring justice to those who have waited too long.
What an incredible case. Hearing from the investigators who dedicated years to solving these crimes really shows their passion and commitment to the victims and their families.
Absolutely, Vanessa. And imagine how many more families could get the answers they deserve if lawmakers across the globe prioritize retroactive DNA collection for violent offenders. As we wrap up the 2025 Hit of the Year, we want to thank all the agencies that submitted cases and participated in this year's program.
And a special thank you to Thermo Fisher Scientific for making DNA Hit of the Year part of HIDS.
One important update, we're changing how cases are submitted. There's no longer a time limitation when the DNA hit occurred. Whether it happened last year or 20 years ago, we want to hear about it. Plus, if your case isn't selected in the year you submitted it, you will remain eligible for future consideration. We hope this encourages even more submissions. Our portal to submit new cases is now opened. Please visit DNAHitoftheyear.com.
Thanks for joining us.
See you next year.